Assalamualaikum warahmatullahi wabarakatuh and a very good day to everyone. Today we are from Group 3 will present an experiment which related to topic tiometric analysis. So the title is Experiments of Acids, Bases and pH by using Red-Cabbage indicator solution. Without further ado, I will start presentation by introducing my group members. They are Audrey Ashlinus Elhin, Sandra Ellen Job, Nur Suhaidanuddin, and me myself Nur Amira Yahya.
Acid-base titration is a technique to determine the concentration of an acid or a base solution by neutralizing the unknown concentration of a solution with the known concentration of base or acid. The example of this acid and base titration that used in real life is analyzing the purity of medicines. The drug ephedrine, for example, is commonly found in the cough syrup, can be tested for purity by using acid-base titration. A pH indicator is used to monitor the progress of acid-base titration. So in this experiment, we are going to use Red-Cabbage as our indicator. The reason that we use Red-Cabbage as our indicator because it contains an indicator pigment molecule called flavin, which is a type of molecule called anthocyanin. Consequently, the color of an anthocyanin solution turns can be used to determine a solution pH, which is a measure of how basic or acidic a solution is. So this is our main journal and mostly of our ideas refers to this journal.
Next, there are two objectives that have been recognized in this experiment. The first one is to determine the pH of common solution. Secondly is to understand the pH difference of acids and bases.
Next is the methodology in this experiment. First of all is by preparing pH indicator from Red-Cabbage. For the first step is a small Red-Cabbage was grated into small pieces and were left into a 500 ml beaker. Then distilled water was added. The mixture was boiled gently about 5 to 10 minutes until the color became dark purple. The heat was turned off and the solution was left to cool. After that, the cabbage and its solution in the beaker was strained into another large beaker, and the purple solution from the cabbage was used as the indicator solution to test the pH of standard solutions. Next is the part A, which is the qualitative analysis for pH values. First, 10 large test tubes were prepared and labeled with the solution to be tested. Next, 3 ml of each solution was poured into the appropriately labeled test tube. Then, an equal volume of cabbage indicator solution was added into the labeled test tube by using a dropper pipette. The mixture was stirred by using stirring rod. The resulting color of sample was recorded after it was mixed with the cabbage indicator, and the color was compared with the pH color to determine the pH of each sample. And the last part is part B, which is quantitative analysis for pH values. First of all, the probe was prepared to take the pH measurements. The storage bottle was removed and the lower section of the probe was rinsed with distilled water. The same 10 test tubes from part A, which contained the same sample, were used. Then, the pH probe was inserted directly into each test tube. Then, the pH value that was shown on meter screen was recorded. Next, the probe was rinsed with distilled water thoroughly after each pH measurement. Lastly, the probe was rinsed with distilled water when all pH measurements were taken.
Based on this experiment, we have two parts for the result. For the part A, it was a qualitative analysis for pH value. In the table, we have a pH standard and the result for color with cabbage indicator. Okay, the value pH standard from 1 to 3, the color with cabbage indicator was red. Then the 4 until 6, the color with cabbage indicator was violet. Then from the picture, we can know that value from 0 to 6, it was acidic, and 7 was neutral, and from 8 to 12 was alkaline. Alright, we move to the part B, which is quantitative analysis for pH value. In this result, we have used a different chemical to mix with the Red-Cabbage juice. The first one is distilled water. After distilled water mixed with the Red-Cabbage juice, the color with indicator is violet, pH standard is 7.94, and it was a neutral. And the second is vinegar. Vinegar has been mixed also with the red cabbage juice, and the color width indicator is red also because the pH standard is 2.59. This is show that the solution is acidic. The third one is baking soda. The color width indicator for this solution is blue, and the pH standard is 8.29. This is show that the solution is alkaline. And the next is 0.1 m hydrochloric acid. This solution, after has been mixed with the red cabbage juice, the color width indicator is become red, and the pH standard is 1.56. This also show that the solution is acidic. Next is cook. After cook mixed with the red cabbage juice, the color width indicator is red, and the pH standard is 2.56. This also show that the solution is acidic. Next is ammonia. The solution of ammonia and red cabbage juice show that the color width indicator has been turned to green, and the value for pH standard is 11.21. This show that the solution is alkaline. And for the liquid detergent, the color width indicator is blue, and the value for pH standard is 8.58. This also show that the solution is alkaline. While the salt water, the color width indicator has been turned to violet after the red cabbage juice mixed with the salt water. It was acidic because the pH standard was 6.31. And the next one is drain cleaner. Drain cleaner also mixed with the red cabbage juice. It was alkaline because the pH standard was 12.49, and the color width indicator is green. The last one is isopropyl alcohol. The color width indicator is violet because the pH standard value is 7.60, and it was neutral.
By boiling the red cabbage leaves, we extracted a class of pigment molecules called anthocyanate into the solution. Anthocyanate molecules will change their color depending upon the pH of their environment and can indicate the pH of a solution. So, the pigment anthocyanate, when it is exposed to hydrogen or hydroxide molecules, the molecules of the indicator change shape and reflect light of a different shape, where for red color, it indicates acidic like vinegar, and green for basic chemicals like sodium drain cleaner. So, why did the cabbage solution changes color? This is due to the pH level of the chemical added that ranges from 0 to 14, indicating whether a chemical is acidic or basic. So, a lower pH value indicates a chemical that is more acidic, and inversely, a pH value that is higher indicates a more basic solution, but a neutral solution has a pH 7. The pH value is determined by the number of hydrogen or hydroxide molecules in the solution, with higher concentration of hydrogen ion for acids and high concentration of hydroxide ion for bases. While for an unknown chemical, a pH indicator is used to change color. A pH indicator is a substance that, when a small amount of it is added to a solution of unknown pH, will change its color. This is a way to determine pH of a solution visually. The indicator used in this experiment will be obtained from a natural source, red cabbage. Cabbage indicator produces a particular color depending on the pH of the solution.
In conclusion, the objective was achieved because the pH of common solution has been determined, and the difference of pH between acidic and base also can know. Red cabbage juice will change its color when an acidic or alkali solution add into it. Red cabbage also can act as a natural indicator.
This is our reference list, including the YouTube video link that we refers to.